This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 17, recorded September 24th, 2010. Hello, everyone. My name is Vincent Racaniello, and across the desk is a sleeping Dickson Despommier. Hello, Dickson.
Oh, gee, Vince, you woke me up.
You know, it's the end of September, and it's 85 degrees here in New York City.
It is.
Which is atypical. They call this a Native American summer.
This was the hottest summer on record so far.
What about off the record?
We don't have records that go that far back.
Hottest summer on record in New York City, really?
Yeah, ever since we've been keeping them, this is the hottest summer.
Is that global warming?
It's hard to say because these could be just perturbations in a normal pattern. For instance, you know, you always get that argument, but I would think that having had the warmest summer ever, preceded by the warmest summer ever, preceded by the warmest summer ever for the last 10 years, that's pretty good evidence that something is happening out there, don't you think?
And what's the effect on the life out there?
It's rearranging the landscape as we speak.
And mosquitoes?
They're either taking advantage of this or not, depending on whether they're warm weather mosquitoes or temperate zone mosquitoes. There are different kinds.
Is it our fault that it's getting warmer?
Hard to know. But I sincerely believe that it is partially our fault.
Well, we're part of the landscape, and whatever we do is part of evolution.
Of course. My take on this is that the climate changes regardless, but the rate at which it changes is probably being driven by anthropogenic activity.
Weren't there previous periods in the history of the Earth when we got very warm and there were polar melts?
Of course.
Yeah, so it's not the first time it's going to happen.
Nope, and that was due to increased volcanic activity. But in this case, it's not due to increased volcanic
increased
activity.
Human activity.
Yeah, that's right, exactly.
So do you think we'll have a polar melt?
We're having one now.
And the tides will go up?
Well, if the North Pole melts, no, because that's all seawater to begin with.
If the South Pole melts?
That's different.
That's fresh water?
All fresh water. And so is Iceland.
So what would
and
that
so
do?
is Greenland.
Why would that have an effect?
Well, that puts more water back in the ocean.
Well, why wouldn't the seawater at the North Pole do that?
Because it freezes. It's saltwater ice. So it's neutral in terms of the level of the ocean.
I see. And how much, if the entire South Cap melted, how much higher? Would everything flood?
Vince, you don't want to know.
So right here would be underwater?
Absolutely.
This building's 17-story building?
Possible.
Wow.
Possible.
I got to get my drobo out of here.
You trade your drobo for a rowboat.
A rowboat. Very good. Are you here every night?
I'm here every night except Wednesdays.
Excellent.
Try the veal.
All right. We've probably scared away most of our listeners by now.
That's right.
So we can stop. Let's go have a wine. All right. Last time we talked about a very interesting but insignificant parasite named Giardia.
I wouldn't put it that way at all.
I just wanted to get a reaction.
Especially if you've had Giardia. You wouldn't call that insignificant no matter what.
I haven't. Well, I might have. It might have been mild. Who knows?
It could have been.
We could tell. We could take some of my serum and look for
We could look for antibodies.
Antibody.
Yep.
But I don't mean to belittle Giardia.
No, please.
Now what will happen is I will get it in revenge.
Or you'll get lots of email, that's for sure.
When we talk about Montezuma's revenge,
we do.
Do we talk about bacterial infections mainly?
It's
hard to say.
A moving target, no pun intended on that one. It could be due to just a change in E. coli, basically, a toxigenic E. coli versus the one you've got. That's called traveler's diarrhea.
Well, it could be just a change in your gut flora.
Yeah, that's the point.
Imposed by the diet.
That is the point.
And you feel bad, and then you get home.
I think it's a change of E. coli, basically, because it's such a common thing for travelers to get. And in fact, they get it when they come here, too. So if you're thinking the only place to go get Montezuma's revenge is where Montezuma used to live, think again, because it happens to those people living in Mexico that come to this country as well. So
does it happen to you when you go to Mexico?
It's happened several times. Not all the time, but it's happened.
What about when you go to Europe?
Rare.
So it's interesting, isn't it?
Rare, yep.
So it must be an equator thing, right?
Or perhaps it's a milder strain, and perhaps there's more social intercourse between Europe and the United States, and therefore there's more exchange of E. coli.
Is Mexico around the equator? No, it's north.
No, it's north.
Where is the equator?
It's way down in South America.
And so it's not an equator thing.
No.
It's just a flora thing.
I believe so.
How about, have you been to China?
Yep.
Did you have a change of flora?
I probably did, but I didn't stay there long enough. I was there for a couple of days. I wasn't there long enough to really know the difference. But I did spend a month in India.
How was that?
Oh, it was fabulous.
You did that. You went away from TWiP for a month.
I did.
Bugger.
Well, I was out gathering data for these broadcasts. No, that's not true. But the point is,
you were having fun.
We were having fun, but we were also giving talks and visiting with architects and things like this. But I never did get sick in India, and neither did my wife. So, mythologies exist all over the place about these things. And so we're about to dispel a few of them. How's that?
What are we going to dispel today?
Well, we began our discussion last time on diarrheal-causing protozoans.
Yeah, protozoan parasites that cause diarrhea. We
have Giardia.
Giardia, which we did last time.
Yeah, well, we haven't completely done it, but we've sort of concluded that little portion of it.
Right.
We're now going to do Entamoeba histolytica, which is another name for it is amebic dysentery.
Amoebic dysentery.
Correct.
Is there other form of dysentery?
Bacillary.
Bacillary
dysentery.
Is there viral dysentery?
Well, we have to define what dysentery is
what
first.
is dysentery?
What is dysentery?
Dysentery.
Right.
Let me guess.
Please.
Bloody, watery diarrhea?
Got it.
Not steatorrhea.
No.
That's what Giardia does, right?
Vince, you've got an ironclad memory. Absolutely
I have an ironclad intestine, too. Steatorrhea.
Well,
bloody, watery diarrhea is dysentery.
Yeah. Whereas just normal diarrhea, of course, is not without the blood. It's without the blood. So
wait. Without blood, it's just watery. And
without the blood, it doesn't have to be watery. It doesn't have to be watery. It could be
loose,
fairly loose.
It's just diarrhea.
Yeah.
So I don't know what the rotavirus is. I know they cause diarrhea. I'm not sure it's bloody. No, it is not bloody.
It's not bloody.
Where does the blood come from? I guess we'll find out.
Salmonellosis causes it.
Mm-hmm.
And Shigella causes it.
All right. Those are bacillary dysenteries.
That's
now
right.
We have amoebic dysentery.
Now we have another one to cause.
So Entamoeba is the only parasite, a eukaryotic parasite that causes dysentery?
As far as we know.
All right.
As far as we know. And it's an amoeba, so it's a member of a class of, an entire phylum of organisms called, and I'll come up with this name in a moment, Vince. I'm having a senior moment.
It's not
You're going to edit this out.
Protozoan?
No, it is a protozoan. Of course it's a protozoan. All of the amoebae that we'll be discussing today have a single nucleus.
Yeah.
But the one that causes the disease that we'll be discussing, called amoebic dysentery, does not have any visible lysosomes, Vince, but yet it causes cells to lyse. So we'll discuss its mechanism
there are many
of
ways to
pathogenesis.
lyse cells.
There are tons of them. Tons of them.
I'm a little confused about the classification now.
You are.
Protozoans, we've talked about a bunch of them so far. We're actually focusing initially here on the protozoans.
Yep.
So I'm opening your book to the table of contents, and everyone should have one now. This book, of course, being Parasitic Diseases, because it's cheap. And
it's cheap. Oh, boy, is it cheap.
The podcast is free. Might as well buy Dickson's book.
Yeah, right.
Protozoa, Giardia, Leishmania, Trypanosomes. We haven't done Trichomonas, but we will.
No, we haven't.
The malarias.
Yep.
We haven't done Cryptosporidium.
Nope, but we will.
Toxoplasma we have done.
Yep.
And then a few others. Then they have at the end Entamoeba. So all of these others are protozoan. They're all protozoan parasites.
Yeah, of course.
But the others are not amoeba.
No.
So what is an amoeba?
An amoeba is an organism that apparently has no definable shape. It can assume an amorphous shape, and it has no visible locomotor organelles. Okay?
No flagella, no cilia?
None.
No,
it moves by
tires.
extending its pseudopod in a particular direction.
Right. So
it's related to the free-living amoeba.
Why is it a protozoan?
Well, because it's a single cell and it has a nucleus.
Anything with a single cell and a nucleus is a protozoan. So
That's correct.
The cells of my skin are protozoans?
Well,
they're not free-living.
They're highly organized into a structure you would call skin. That's right. So they're not free-living.
But to be free-living, that's the key?
That's right. And
Do these guys make biofilms?
Make them, or do they depend upon them?
Well, as we know now, we understand that bacteria make biofilms. They live in communities.
You're right.
And they interact.
You're right.
They
secrete
planktonic
polysaccharides
growth,
and stuff. Yeah, that's right.
In single-cell suspension, which is what we do in the lab, is not the norm out there in nature.
No, of course not.
So my question is, do the protozoans make biofilms? Do they form biofilms?
It's a superb question, but no, I think they take advantage of biofilms rather than form them themselves. So all of these protozoan parasites, and including Entamoeba histolytica, and I am glad you should ask that, of course, because Entamoeba histolytica has a particular way of attaching to cells, much the same as we discussed in some of our earlier This Week in Virology programs. There are receptors and there are ligands that come together to enable the organism to remain in contact with its target cell. And we'll discuss this.
All right. Now, this guy, Entamoeba, apparently there are different species. We have Histolytica.
We do.
We have Dispar.
Right, which creates a conundrum among the diagnostic technicians because they're impossible to tell apart by looking at them under a microscope.
Why would you want to tell them apart?
Well, because somebody might be suffering from it, and they might submit a stool sample for examination, and you might want to know whether it's being caused by Entamoeba histolytica or some other organ.
Why would you want to know?
Because if Vince,
don't you treat them the same way?
Of course not. Absolutely.
But I'm leading you on, Dickson.
You are, Vince. as you do. No, all of these diarrheal disease-causing agents require different modalities for treatment.
Okay.
So that's why you have to know first.
Entamoeba histolytica was the first discovered?
Was discovered of the diarrheal disease-causing agents?
No, of the entamoebas, of the amoebas. Amoebas.
It's a good question. I mean, who first saw the first amoeba? All right. Dobell was the discoverer of Entamoeba histolytica. So Dobell is the guy giving credit for this. But there are some other people also. You can see their names associated with Entamoeba histolytica that have nothing to do with Dobell. But Dobell was a great microscopist who also was very curious about
roughly what year?
Pathogenesis back in the 1800s.
Now, Leeuwenhoek. I always get it wrong.
That's all right.
Leeuwenhoek.
Yeah.
He didn't see amoeba?
Apparently not. I mean, in that little hanging drop preparation, it would have been very difficult to find out what that was. What he saw, though, were these little leaf-like organisms, which he described, and which we now know as Giardia. Yep.
So they have a very defined morphology.
Very defined. But whereas these
amoeba
don't.
are amorphous. Pleomorphic, right. We would say, if it were a virus.
You're absolutely right.
All right.
That's right. So there's a whole bunch of free-living amoebae. In fact, if you go to the literature on this, you'll find out that the majority of flagellates and amoebae are free-living. They're not
But those are not all parasites.
They're not all.
This one are parasites because they require a host.
That's correct. That's the definition of parasite.
One of the hosts is us.
That's right. In fact, the Entamoeba histolytica is quite specific to humans and to primates in general, and mostly higher primates, not the lower primates like lemurs. Mostly apes, the great apes. And
no other animals in general? You
can force them into infections by doing certain things. All right. But for the most part, in nature, there are no reservoir hosts for this.
So this is different from Giardia, which live in beavers, among other things.
That's right. That's exactly
What do they call the Giardia again? The beaver what?
Beaver fever.
Beaver fever. Why didn't I know that?
I don't know, Vince. There's a lot to know about these subjects.
So why did we discover Entamoeba histolytica?
Why?
Yeah.
Well, we were seeking a cause for a particular clinical syndrome.
Dysentery.
Yeah.
Where and when?
Back at the turn of the century, basically, when all the microbes were being looked at and questioned as to what their role in nature was. And lo and behold, back in the 1910s and 20s and 2030s, it was definitively shown that an amoeba that could be identified by staining characteristics as a single nucleus also possessed in the cytoplasm of the trophozoite stage, and now we have to talk about the life cycle, contained red cells in many cases. Now, it ate living cells, okay? Is a red cell a real cell, Vince, or is it just sort of like an almost real cell?
Well, I wouldn't say real because it doesn't have a nucleus.
That's correct, but it did at one time.
It did at one time. It was ejected like a ejection seat. And,
exactly.
It's left with a lot of mRNAs encoding globin.
That's right.
Which is what it needs to carry oxygen, right?
That's correct.
And that's why some of the first mRNA studies were done on red cells, because it gives you almost a pure population of one species of mRNA.
That's exactly right.
So before cloning, it was the only way to do that.
So if Entamoeba histolytica feeds on red cells, and if that's all they ate, would that actually be a parasite?
It would be a predator. A predator eats all of you.
Predator hunts you down.
Yeah, it hunts down, kills you. But if it ate just red cells, sure, why not?
Well, are red cells cells? That's the point.
Well, they are cells. They are.
What's the definition of a cell, Vince?
A virus.
No, stop it. Now we're really getting off on a tangent.
Well, you say, what's a virus? So
I'm raising a question here because...
What's the definition of a cell?
Yeah.
Let me Google it.
I don't think you're going to find this. I don't think even Robert Hooke could have given you a good definition of a cell.
A biweekly publication of research.
Right.
That's the first thing that comes up, which is really tragic because it shouldn't be. Cell is the functional basic unit of life, discovered by Robert Hooke.
That's right.
And a functional unit of all living organisms. So whether a red blood cell is a functional unit, well, it is. It serves a function.
It does.
It's a bit crippled. It can't divide. But division is not part of the definition, is it?
That they didn't define it that way.
It's the smallest unit of life that is classified as a living thing. Oh, that excludes viruses, doesn't it?
I'm afraid so, Vince.
David Baltimore said viruses are alive, in his opinion.
Did he say that?
Yeah.
Well, who's going to argue with the Nobel Prize winner?
Well, I think arguing is the point, not what the answer is. And he said it doesn't matter whether they're living or not. But
they
he
exist.
agreed that debating it was fun.
Right.
He said he loved viruses.
Apparently so.
Some organisms, such as most bacteria, are unicellular. Others, such as humans, are multicellular. So they say it's the smallest form of life. This is Wikipedia, but I would debate that definition.
It's got lots of exceptions, and the red cell might be one of them.
You know what the largest known cell is?
I think it's a brown algae.
It's a ostrich egg.
Oh, sorry.
3.3 pounds.
Sorry.
It's the unfertilized ostrich egg cell, which weighs 3.3 pounds.
That's an animal. What about a plant cell?
Well, that I don't know.
I think the largest single cell is a brown algae.
So are we debating whether Entamoeba is actually a parasite?
Well,
it's in your book on parasitic diseases.
Wait a minute. If it only ate red cells, then
you wouldn't say it was? What would you call it then?
You might want to rethink that problem, the same as what you would talk about with regards to malaria.
So what would you call it if it just ate red blood cells?
Oh, and I needed those red cells. Boy, it was still limiting my resources, and it's still a parasite in that sense.
It is. It's taking your globin and...
That's right.
In fact, hemoglobin, right?
Inside
there, and it's probably making you pale.
That's true, or it's certainly inducing more red cells to be formed.
Yeah.
Now, this organism, Entamoeba histolytica, causes GI bleeding by eroding away the lining of the large and small intestine, mostly large intestine.
See a picture of it here in your life.
It causes something called craterform ulcers.
Wow.
And they're horrible, horrible things for someone to have. Yep. They can often be mistaken for other entities that cause similar but different clinical conditions. The most confused one is ulcerative colitis, which has bloody diarrhea associated with that as well.
Not caused by a parasite.
No.
According to your book, Dickson.
Uh-oh. I'm going to end up eating my own words here.
No, it is... No, you're not eating your own words. It says it was described in 1875. Clinical features of infection with E. histolytica reproduced some aspects of the disease in dogs. This was Loesch. And then in 1893, E. histolytica was distinguished from E. coli, Entamoeba coli.
Right.
A non-pathogenic amoeba.
Right.
Often found in normal stools. In 1903, Shodin described the trophozoates and cysts of E. histolytica.
Yep.
He died at the age of 35 of overwhelming amoebiasis. Amoebiasis? How do you say?
Amoebiasis.
A tragic outcome of self-experimentation.
A lot of people did that to themselves during those times.
And then some other studies. Dobell's 1928 fully elucidated its life cycle.
Dobell was... The guy that's credited with the life cycle is correct.
What fraction of human dysentery is caused by E. histolytica?
Depends on the part of the world you're in.
Okay, let's take some surveys.
You go to Durban, South Africa, for instance, or if you go down to Mexico, it's Possible that it constitutes as high as maybe 10 or 15%.
Did you say Mexico?
Yeah.
Oh.
About as high as 10 or 15%. Because it's most common, as you know, Vince, to have rotavirus or salmonella or shigella. Those are easily transmitted from person to person. This one requires several manipulations of the environment, both in the large intestine, and also it depends on fecal contamination, just like the others, by the way. So there's no difference in their transmission zones. There's no vectors for this, by the way. So
vectors.
Entamoeba histolytica is acquired through the ingestion of a cyst.
And we are the definitive hosts.
And we are the definitive host. But there's no sexual stages to this, so there's no real definable definitive host. This doesn't undergo sex.
We're the only host.
Well, like I said, higher primates can also catch it, and they mentioned that dogs could be experimentally infected as well, but not common.
So it just does binary fission.
Yep.
Not sexual reproduction.
That's it.
Okay.
Sounds pretty simple, right? Sounds like Giardia, basically. In fact, it has a lot in common with Giardia in terms of its life cycle.
So should we do that now?
Sure.
Are we ready?
What
does it look like?
Well, it varies in shape because it's an amoeba.
But the infective stage is
the cyst.
This quadranucleate cyst.
That's correct.
Which means 4 nuclei.
And Giardia has 4 nuclei in its cyst too. But remember, the trophozoite of Giardia has 2 nuclei.
Yes.
Whereas the trophozoite of Entamoeba histolytica only has 1.
1. And then...
So what this does is it jump starts the infection. For each cyst, you get 4 trophozoites that come out. All right, so it's got a multiplicity factor.
So you eat
here.
it. And
you eat it, you ingest it.
How do you eat this thing in contaminated food?
Yeah, it could be from a food handler that didn't wash their hands before they came back out into the main dining room to serve you your salad that they tossed in front of you.
With their hands.
With their hands. It could be a food handler working in the kitchen.
Would cooking the food kill it?
Sure.
So if it's cooked.
That's right.
But it's not something you would get from beef.
No.
Like some of the other parasites.
Nope.
It's from people. Person-to-person transmission.
It's from people. Or fecal contamination of water. It's very common, by the way.
I see. Runoff, sure. So if I go camping and the water is contaminated with sewage, I could get it.
You could.
Along with Giardia from the beavers.
That's right.
Or other humans. It says here 50 million people are infected worldwide. That's not a lot.
But 50 million compared to 6.8 billion.
There's nothing.
But if you have it, it's something. And we're always on the lookout for it.
I heard a guy talk once about listeria, and this is a little rat hole, but it's worth mentioning. Mm-hmm. He said, if you get listeria, you don't die, but you wish you would.
Right. Right. Well, people have actually died from this one.
Really? So you can die of Entamoeba histolytica infection?
You can, but it's usually not from the intestinal form that you die from. So this parasite has another trick up its sleeve.
Sleeve?
I
thought it was an amorphous amoeba.
Stop it. This protozoan.
You want me see? This is an edutainment thing here.
I agree with you. Okay, he's baiting me again, folks. But the point is that Entamoeba histolytica is an obligate anaerobe.
Ah.
And so is Giardia.
Giardia was as well.
They're both.
If you give it oxygen, it's toxic, right?
Well, or they don't divide, or they don't feed. They just sit there and wait for that to go away. But this parasite, this one in particular, unlike Giardia, can invade the tissues. And in fact, like its name implies, Histolytica, it lytics histos, it lyses tissues.
So that's where the name comes from?
Correct.
Excellent.
So it actually can invade the tissues and get involved in the blood supply. And from there, in the large intestine, the blood supply could carry it back to the liver.
Mm-hmm.
Now, it has some, nothing has choices because they don't have a brain, right? But the point is that they could stick in the liver and begin an infection there, that's the life-threatening aspect to this.
So you ingest the quadranucleate cyst. They go to your intestine.
To the large intestine.
And then they multiply?
They attach to the columnar epithelium.
Okay.
By a special mechanism that's talked about over here that requires a certain
kind of sugar, right?
Sugar, that's right.
And then they become trophozoites?
That's correct.
So one quadranucleate cyst becomes 4 single nuclear trophozoites.
That is exactly correct.
And those enter the circulation?
No, not the circulation. They attach to the gut tract.
They stay right there?
And they start to erode. They suck in that columnar cell, they digest it, and at the same time they're sucking in another cell, so they're greedy little sons of guns.
Why are they doing this?
To feed and divide.
Are
They're
they
dividing and conquering.
They're feeding on your tissue?
That's right.
As well as red blood cells.
Well, they haven't gotten to the red cells yet. You see, they have to get down into the submucosa.
How do they do this? Do they have enzymes that digest?
They do. They have tons of them, and they're all acid hydrolases. But like I said earlier, this organism, if you look at them underneath the electron microscope, you don't see any lysosomes. So where are these acid hydrolases coming from, Vince? And the answer is that they're made on contact. As soon as this organism contacts a cell, it begins the process of making these acid hydrolases. And not before.
In the cytoplasm?
Right, at the contact
But they must be at the contact point.
Yes.
Which is a membrane, a lipid membrane.
Yes, and it also produces one other kind of protein. It's called a pore-forming protein. And that protein can actually poke a hole into its target cell and cause a leak to develop. And when that happens, of course, the cell dies. And the organism then takes
I see an interesting conundrum here, how the amoeba doesn't digest itself. Right?
It's like a poisonous snake. Why don't they die from their own poison?
Exactly.
There's a bunch of interesting philosophical points to discuss, but I think we should get on with the life cycle instead.
So we're in the intestine. We're stuck on the columnar epithelium.
Then we digest our way through that layer.
Yes.
And we get into the lamina propria. And from the lamina propria, the organism has a way to actually burrow down through the basement membrane and get involved in the submucosa, which is where the muscularis is and where the circulation is.
And lymph and capillaries.
And once that happens, the organism has the option of entering the blood supply. But out come the blood cells at that point. When they digest their way through a capillary, for instance, the capillary starts to bleed. And the organisms start to ingest the red blood cells. And that's when the patient starts to develop dysentery. It could start out as diarrhea, but it could end up as dysentery.
That's where the blood is coming from.
That's right.
Now, the trophozoites are smaller than the diameter of the smallest capillary? No.
Oh, no. They're quite large.
So they need to get into a lymph capillary or something larger.
An arterial.
Or
a venule.
Okay.
Probably a venule.
So they keep chewing away till they get to one of these things.
They do. And then by accident, the circulation picks them up and away they go. And they end up in the liver. 99% of the time they end up in the liver, but every now and then they can go bypass the liver and end up in the brain.
Yeah, well, not every one will stay in the liver. The liver filters out bad things.
This is true.
Reticuloendothelial system. And in fact, the circulation from the intestine goes right to the liver.
And why is that, Vince?
Well, you digest your food there.
And what else?
What else?
Yeah, what else?
Wine. I don't know. What else?
No, the alcohol is actually absorbed in your stomach.
Is that right?
Yes, that's true. That is true.
I didn't know that. And so what?
So to avoid getting drunk, you swallow a little olive oil first, and it coats your stomach, and then you can drink all you want. It goes down into your large intestine. Nothing happens to you.
It's an old wives' tale.
No, it's true. It's true.
How much olive oil do you have to drink?
Not much.
Well,
okay, so that wears off, by the way, after an evening of ribaldry.
Why does the circulation go from the intestine to the liver?
Well, there's another function that the liver performs besides removing particulates. It detoxifies. Where are those toxins coming from, Vince?
Your food?
No, they're coming from your bacteria. These are endotoxins.
Oh, so your bacteria are metabolizing some things that you eat and toxins go. What kind of toxins are we talking about?
Well, E. coli produces a very potent endotoxin. So the liver, through their P450 enzyme systems, digests and gets rid of all of those toxins. There's a lot of different P450s, right?
Yeah, there are.
So the point is that's what the liver does for a living. And yet this parasite seems to be able to invade that space. Now, it also requires living cells. This parasite will not feed on dead cells.
Well, we don't have any massive amounts of dead cells in us, right?
We don't, but I can tell you now that this parasite actually creates a lot of dead cells.
So we're in the liver. Okay. So we got diarrhea.
We do.
Bloody diarrhea. And then we
got
always
dysentery.
go to the liver, or we...
Not always, but there's something unique that happens when the parasite becomes extra-intestinal, as it's called.
Yes.
And if it gets to the liver,
right,
then the gut tract infection usually goes away, which is an unusual feature of this. So you might not suspect this parasite as to causing this amoebic abscess in the liver Because you'll say, well, if it's caused by Entamoeba histolytica, that's a gut tract dweller. Yeah. So I'll look for it in the gut tract, and it's usually not there by the time it's become extraintestinal. So that confuses the diagnosis. But there are some modalities here that you can take advantage of. Serologies.
Is this abscess in the liver a problem?
Vince, I've been told, although I don't have evidence directly, that this is a very painful episode. And by palpating the liver, you can actually cause the patient to jump up off the table, basically.
What side of your body is your liver on?
Well, it's all it's all the way underneath your costal margin.
Well, the big part, the big lobe, is
that's the right side.
But you could palpate it across the.
If you hit that abscess on the right side of the liver just right by palpating it, the patient will just scream bloody murder because it's a very, very painful event. There are other kinds of abscesses in the liver that are not painful. Hepatoma is one of them. Okay, so that's not a good way to differentiate these, by the way.
Yeah, sure.
So the point is, and by the way, notice that there's a little bubble of abscess. Yeah. That's very typical for this infection. Now, what's on top of this, do you think, in us?
Diaphragm?
That's correct. If this ruptures, this organism has the option of eroding its way through the diaphragm into the lungs.
So it erodes wherever it is.
It eats living tissue.
So this will likely rupture because it's going to erode through the abscess, right?
Well, yes and no. It can smolder for many, many months, basically. It can smolder.
From the time you ingest to when it starts eroding your intestine, how long does this take?
That could be a week to 2 weeks.
And then we're weeks in the liver abscess. Yeah, and that
weeks to months.
That stops the multiplication of the colon.
Or the organism suddenly disappears from the colon for some unknown reason. I mean, there might be some feedback. There might be some molecules that this organism actually secretes that says you can't live in two places at once. We don't know this. The quorum sensing molecules that
and you were excreting trophozoites.
Yeah.
In your feces?
You are, but actually...
Or they become cysts?
They round up and become cysts. That's exactly what
quadrinucleate cysts?
They, first of all, start as a uninucleate cyst.
And then they divide twice.
They divide twice, that's right.
They become quadrinucleate. You shed them, and then if you're sloppy, you contaminate other people.
Got it. It's pretty simple stuff, but it took a long time to discover this.
Can you get this by stepping on it?
No. No.
You have to eat it.
Although, if I say no to that, the answer is...
Not likely.
It's 99% unlikely.
I guess if you had a cut on your foot.
Exactly. That's exactly
So
right.
you could get it via the blood. It would circulate. It would stick in your liver and multiply and chew its way to other organs. So it could go through your diaphragm.
Yep.
It could cause some lung damage.
It's true.
You could be coughing it up.
Oof, yes. But
bloody cough.
At that point, things are getting pretty bad.
Could go to your brain.
At that point, you're going to die.
So when you die, it's from a brain abscess?
Not necessarily. You could die from overwhelming gastrointestinal infection also.
So what fraction of infected people die?
Not many. This is not usually
I bet
fatal.
If you had AIDS, it would kill you.
Well, at the beginning of this epidemic, it was...
The AIDS epidemic?
The AIDS epidemic.
Yes.
This was known as gay bowel disease.
Gay bowel?
Yeah.
Really?
Because this was a commonly transmitted infection through fecal contamination of oral sex and anal intercourse.
Yeah,
which
right.
Gay populations practice, and in fact,
partners, yeah, sure.
Heterosexuals do too, but it was a very common practice in those days, and it probably still is among the gay community. So it was being overdiagnosed though because of the look-alike amoeba, Entamoeba dispar. Now, Entamoeba histolytica and Entamoeba coli are easy to tell apart because of their nuclear structures. They're totally different. But Entamoeba dispar and Entamoeba histolytica are almost impossible. They can't be told apart. Unless, of course, the Entamoeba histolytica trophozoites that you're looking at have red cells in their cytoplasm, because Entamoeba dispar doesn't take them up. In fact, they're not a parasite. They're a commensal.
What does histolytica do with the cells?
It digests them.
Okay.
And uses the resulting amino acids for its own metabolism.
Did you tell me what fraction of people die? I don't remember.
Low, it's about 5%. But in some areas of the world, like, again, Durban, South Africa, seems to have a high propensity of amoebic abscess.
Now, I presume the cyst is just a resistant form that has to pass through your stomach, right? And
yes.
The trophozoite can't do that probably, right?
No, that's correct.
And so it becomes a trophozoite in the colon.
Got it.
So fecal oral contamination, person to person, humans, main hosts, can make you very sick, can kill you.
This is true.
and anything else.
To diagnose it, of course.
How do you diagnose it?
Well, we should talk about the mechanism of pathogenesis here, too, Vince, because it relies on the certain sugar residues that are decorations for proteins that sit on your columnar epithelium.
It's this attachment step you're talking about.
That's right. It's actually dependent on galactose. So galactose is
N-acetylglucosamine and N-acetylgalactosamine, and
That's
at
right.
least 2 surface lectin proteins. Would you care to tell the listeners what a lectin is?
A lectin, I presume, will relate to the parasite side of things. It's the molecule.
It binds sugars, doesn't it?
That's right. So it's the parasite.
Oh, it's the parasite that's binding the sugar.
The parasite has a receptor for the sugar molecules that are decorating host proteins. And it's a lock and key type of thing, a very simple. We imagine this as a very simple lock and key kind of arrangement of stereochemistry, where the correct sugar conformation allows this parasite to stick, and it won't come loose. And in fact, that event triggers the synthesis of the acid hydrolases, which then end up and the pore-forming protein, which then kills the host cell. So you can incubate a bunch of amoebae in a cell culture containing, in this case, Chinese hamster ovary cells. And as long as that sugar is present in solution, galactose, you can prevent the amoeba from attaching to its host cells. And they will live side by side. I call this Daniel in the lion's den.
So the amoeba are living near the cells but not attached.
They don't know that they're the host cells because the receptors haven't been encountered, because the solution is flooded with the receptor and binds the lectin on the surface of the parasite.
So the lectin is on the parasite.
It is. And the moment you take the sugar out of the solution,
kill the cells.
They immediately detect the sugar on the surface of the Chinese hamster ovaries.
And they put all these enzymes in that kill the cells.
They kill the cells. That's right.
Do these reproduce in culture?
Sure.
I see here that there are also viruses of E. histolytica.
There are, Vince. There are. And Lord knows what they're doing. Are they important for virulence factors?
It depends whether it's killing them or... Oh, look. One virus has no effect on certain strains of amoeba.
Right.
But can kill others. So in some it maybe transfers genes among them, and others it kills
Is that
them.
a paper by any chance by C.C. Wang?
Oh, was CC the guy who worked on this?
Yeah.
I interacted with CC many years ago.
Yep, he's
I used
still
to
active.
handle his papers.
Oh, really?
I think he also worked on Leishmania viruses, too.
He did, that's right. He likes to work on unusual and biologically interesting.
There's also a lot of space in these unusual areas. There's not a lot of competition,
That's also true.
I mean, there's nobody pays attention, but there's also not
until they become important, until you find out what their function is, and the next thing you know, everybody's working on them. That's right. So parasites have a lot to teach us, and this is a clear-cut case of that little limerick that I read to you very early on in our series about little fleas have smaller fleas.
By the way, C.C. Wang's daughter married one of our former students.
Is that right?
Yeah.
Well, C.C. and I served on the same study section together at NIH, so we've had a nice encounter. He's a very interesting guy, very nice man.
C.C. Wang. C.C. Good to have initials C.C. Hey, C.C.
We used to call him ML.
Why?
Well, C.C. stands for cubic centimeter.
Oh, you called him ML?
That's right, for short.
Well, there was a virologist named Roland R. Ruckert.
Yes.
Do you know what we called him?
I have no idea.
R cubed. That's really geeky.
That's geeky. Well, my friends call me D cube.
DDD. Dickson D. Despommier. D cubed.
That's right. D stands for Donald.
Donald? I didn't know that.
Yeah, well, you never asked me.
Hey, Don.
No, don't do that.
You don't want me to call you Don?
No, not at all.
All right.
No, I prefer Dick or Dickson.
So these attach via lectins to surface sugars.
That's right.
And as soon as they find that surface sugar, boom. According to your chapter, Dickson.
Game over. Game over. Yes, Vince, you're going to make me eat my words again?
No, not at all. I'm just providing some detail.
Okay.
The surface membrane of the trophozoite contains phospholipase A.
It does.
Which will chew up lipids.
That's another acid hydrolase.
Neuraminidase.
That's true.
Which will cut off sialic
acids. We know about
acids,
neuraminidase, yes.
And a metallocollagenase. That's a collagen-cutting enzyme that needs a metal.
That's right.
In addition, it secretes a minimum of 4 cysteine proteases.
Look at that.
help it move through the extracellular matrix.
That's right.
This guy has adapted.
Oh, big time.
Now, diagnosis cannot be done by microscopy, right?
Well, you can narrow it down to Entamoeba histolytica or Entamoeba dispar.
But you want to go further because the drugs are specific?
Well, Entamoeba dispar won't go away with this drug, you see. So what you might suggest, if that happens and you've treated with the right drug, is that it's a drug-resistant form of Entamoeba histolytica, which they used to call it. But it's now since been discovered that there are these two organisms that are identical in morphology but quite different in terms of their isozyme patterns. That's the way it was originally looked at by this guy Sargent over in England. He actually cultured both of these things out from patients that had quotes-unquote drug-resistant Entamoeba histolytica and drug-sensitive Entamoeba histolytica, and he found that there was a huge difference in the isozyme patterns. They said, well, these must be two different organisms then. No one believed him. He did all of the work and finally proved to the world that indeed those were two different organisms. And now we have a PCR test for it.
All right, so it's diagnosed. If you displayed symptoms, bloody diarrhea, they would take a stool sample and do PCR.
Yeah, but it could be doing something else too, because if you've had fecal contamination, think of how many other organisms that can cause dysentery.
What's the steps involved in diet? You go to your physician, you have bloody diarrhea, what do you do?
So then they take a stool sample and they divide it, and they take half of this and they culture it. They look for all of the bacterial diseases that it could be. And at the same time, they send it off to the microscopy laboratory for diagnosis for E. histolytica. Well, if they come back with a diagnosis of no cultural organisms that caused dysentery were found, and E. histolytica was seen, trophozoites seen,
right?
And if the trophozoite has red cells in it, then you know that you can be treated with metronidazole, which is the drug of choice, and it treats all anaerobics, not just amoebae, also bacteria.
Didn't it treat Giardia?
It did, Vincent. It's an anaerobe too, remember?
So when you were working in the parasitology lab, did you ever see trophozoites with red blood cells in them?
Tons of them.
Really?
But every now and then you'll see a trophozoite that has no red cells in it whatsoever. Now, is that E. histolytica? Just because the nucleus looks the same? Or is it E. dispar? So in those days, we didn't know.
You didn't have a way to distinguish.
No, we didn't know.
But now you would do a PCR?
Oh, we would do a PCR, sure. But there are also antibodies that you can use now, too.
Sure,
you can
stain them?
You can. You can do an ELISA test for this now.
So if you got the test back, it's not E. histolytica, it is E.
dispar.
Dispar.
Then you have to look for another cause for the dysentery.
Ah, because that doesn't cause dysentery.
Right.
But would it be just there coincidentally with dysentery?
It's a fecally contracted. You can contract this by fecal contamination.
But it doesn't cause any disease.
Correct.
So we could have it right now.
We could.
Is it stay with people forever and ever, or is it just a transient?
It's a good question. I don't know about forever and ever, but I know it's a long-term exposure and long-term association. There's no harm in it, you see. So therefore, it's like a commensal. They call these commensal associations.
Interesting.
It does you no harm. You do it no harm. It lives in your large intestine. It's eating stuff that you would have discarded, namely feces. So who cares? They can have all they want.
It doesn't eat red blood cells, then? Doesn't chew away your intestine?
No, it has no pathogenic.
Does it attach to the wall?
Nope.
It just kind of sloshes around there.
Yeah.
And it divides in your intestine?
Yep.
By the way, how do these guys divide?
By binary fission.
So starting from the trophozoite on the wall, they just divide, and then eventually...
They eat, and they get big, and then they divide. They eat, and get big, and then they divide.
What triggers them to become cysts?
Perhaps there is a quorum-sensing molecule involved in that. I would venture to guess, even though I don't know that for sure, that encystment is a population-dependent phenomenon. And it's the way the organism says, I've got enough trophozoites working for me here. I've got to start infecting another host. So they start producing cysts. I'm sure there are physiological conditions that trigger it, but that's dependent on how many parasites there are. There's a picture in the text of a woman who was diagnosed with ulcerative colitis, but that's not ulcerative colitis. If they had done the sectioning correctly and looked for the organisms, they would have found them. But ulcerative colitis is much more common than Entamoeba histolytica infection. So to make a diagnosis based on symptoms is a big mistake, Vince. You need to do the definitive diagnosis.
You have to do lab tests, right?
You got to do the lab tests.
Now,
that's the big lesson here.
It says here, many infected individuals are asymptomatic and go on to become carriers.
This is true, Vince. This is true for a number of other dysentery-causing agents too, like salmonella, for instance.
Is that
you
right?
can become a carrier for salmonella,
yeah.
right? Typhoid Mary.
Yes.
Remember?
So we have Entamoeba
Amoeba
Joe.
Annie and Typhoid Mary.
Amoeba Annie, yeah. Did you know that the genome has been sequenced?
I did.
It's only 20 million base pairs in length.
Go figure.
Interesting. And it has 8,160 predicted genes.
Very interesting indeed. We should have been able to get rid of it knowing that, but of course, you have to still know the details, don't you?
Should I worry about Entamoeba histolytica?
For the most part, no.
So in New York City, do we see it?
Yep.
Is it a reportable disease?
No. It's rare. But there have been some notable outbreaks.
Tell me.
One of them occurred in the 1893 World's Fair in Chicago, where the freshwater pipes and the sewage pipes had rusted together. This is the seminal...
Why were they next to each other?
They were, because in those days, Vince, they didn't realize what they were doing. And so they dug a trench and they put these cast iron pipes, one carrying the fresh water, one carrying the sewage. So in the fresh water, you got just a little bit of sewage in the fresh water. In the sewage, you got just a little bit of fresh water in the sewage. So what happened was that they had this World's Fair, of course, and it was an expo, and 400 people in one hotel died of the infection.
Died?
They died.
How many were actually infected?
That's a good question.
More?
Thousands.
Wow.
Thousands. This was a very virulent form of it.
Any more recent?
Oh, well, when I was a technician here, and that goes back to the 1960s, we had an outbreak at an orphanage in New Jersey. This is an institutional outbreak organism as well. And all of the slides that we currently have available for showing to our medical students, which we're no longer doing, but which we did for many years, were all derived from this one outbreak out in New Jersey someplace. And the stool samples were teeming with amoebae, and all the amoebae had red cells in them. In fact, all the pictures in this book are derived from those cases. And there's one right there.
That's a trophozoite.
It's a trophozoite with red cells being partially digested in its cytoplasm. And you've got this little shadow of something right over here.
Yes.
On the next page, it shows you what that is. It's a Charcot Lyden crystal. So what is a Charcot Lyden crystal? It's one of the granular proteins of eosinophils.
Isn't Charcot-Marie Tooth the disease that you picked the paper from years ago on TWiP?
It
Remember?
is. Yeah, I did
so
pick
they had identified genes involved in it.
That is correct.
Is this the same Charcot?
It probably is.
And Lyden is of the Lyden jar? Lyden?
Probably.
They're crystals.
They are crystals. And these crystals are characteristic of GI bleeding with an allergic component to it. So the eosinophils are involved in this. So a long-term association of this infection, by the way, in some patients, very few, and probably there's an immunogenetics to all of this, is the creation of a large space-filling lesion, which often resembles colonic cancer, but which isn't. And it's known as an amoeboma. And the amoeboma contains very, very few living trophozoites, tremendous amounts of cellular debris, and lots of eosinophils. Now, this organism, by the fact that it secretes some of these enzymes into the milieu, secretes a leukocyte toxic agent. So it's very difficult for you to mount an immune attack against this infection because the substance that this protozoan secretes into the milieu of its tissues actually kills off the effector mechanisms for processing antigens and actually getting back into the organism and killing it.
So you don't make antibodies against
You do. You make tons of them, but they're ineffectual.
Right. Okay. But they can be used for diagnosis.
They are very useful for diagnosis. In fact, if you do the ELISA test for this, or there's lots of other things that you can use too. The indirect hemagglutination test was an older test for this. If you're positive for that test, by whatever serologies you choose to use, it's probable that 95% of the cases are diagnosable this way. All right. In 95% of the times that it's positive, you have Entamoeba histolytica.
Have you ever had it?
No. Now, there's a nuance to this too. So Vince, how do you diagnose the extraintestinal form?
You mean the liver abscess?
Yeah.
And the brain abscess?
Because you can't look at the stool anymore because the stage for that is gone, usually.
So you don't have
now
bloody
what do you
diarrhea.
Do? Nope.
But you may have liver pain.
You might.
Or you might.
That's a hint that you might be okay for this diagnosis, but it doesn't definitively.
You go to your physician, you got to say, hey, 2 weeks ago I had bloody diarrhea.
So what if they biopsied it?
You probably wouldn't see very many Entamoebas in there, right?
Where would you see them, though? So you've got something inside of this this abscess called anchovy paste.
Anchovy paste?
That's the name of the...
Are you serious?
The pathology people refer to things by food.
They have a bad sense of
They've
humor.
got a horrible... They must have a terribly boring life eating white bread and water. What else would you eat after all of this, right?
Wow.
So it's called anchovy paste, and it's the remnants of all the parenchymal cells that have died as a result of exposure to these proteins that these parasites secrete to ward off the evil spirits, so to speak, for them. They'll kill innocent cells that are not immune cells also.
Right.
So parenchymal cells fall into that category. Mm-hmm. So when you do a punch biopsy, mm-hmm. and you're looking for the causative agent for this,
right,
and you go through the wall of the cyst,
into the anchovy paste.
Into the anchovy paste, and you draw some of that out as best you can.
Yeah. And
you look under the microscope, you will never see this organism. And that's because the organism requires living cells.
Yeah.
Okay.
Sorry. I made some humming because I'm fooling around with the wires here.
Vince is getting bored. He wants to go home.
I'm
it's
getting
Friday
bored.
afternoon.
I can see that you didn't have enough cable. You want to move around a little.
I'm
there
fine.
You go.
I'm
you
fine.
got
I'm
cable.
Fine. Thank you. All right. So
I never get bored. I'm fascinated by biology.
Okay. Well, this is biology here. So I was disrupted. Where do you want me to pick this back up again?
You put the needle through.
Oh, yeah. So the surgeon that's going to make this diagnosis or going to help the laboratory make the diagnosis inserts the needle as a biopsy procedure. Right now, let's put it this way. If it were a hepatoma, you could make the diagnosis this way, right?
Sure.
If it's a bacterial abscess of the liver, you can make that diagnosis that way too. Mm-hmm. But there's this other thing. Remember where we discussed hydatid disease?
Yes. What
happens when you rupture that one?
Oh, you release everything.
It spreads all over the place.
It's terrible.
So what if this is being caused by hydatid?
By, what was the organism there?
It's Echinococcus granulosus.
Echinococcus.
What would happen to that patient? They would get worse, not better, just because you're trying to diagnose it, right? So what are the criteria for diagnosing by biopsy? Well, if you press on it and it doesn't hurt, it's probably not E. histolytica.
Right. Oh, your tumor, you said.
Or Echinococcus. On the other hand, if you press on it, the patient jumps over the table. You got to know.
Then you do your biopsy.
So wait a minute. You know what they've done in the past? They've said, we're not going to biopsy you. We're going to assume that this is a bacterial abscess. Mm-hmm. Because it's common in liver.
And they give you antibiotics?
No, they give you metronidazole, which is a treatment for anaerobic bacterial abscesses. Mm-hmm. And guess what? You treat the amoeba because it's susceptible to the same drug for the same reasons. That was an older way of telling. Now we can do a PCR. We can do an antibody test and definitively prove, exposure, okay? And if it's exposure with a liver abscess that's painful, it's probably going to be Entamoeba histolytica. Now, Vince, you don't just treat with metronidazole if you've got the intestinal infection. I didn't mention this, but there is another drug that you have to give. Mm-hmm. You give metronidazole, sure, but there's this other drug called Iotaquinol.
Would that be a luminal amoebicide?
It would be a luminal amoebicide, as a matter of fact. There are other names for this. Diiodohydroxyquin is another name for it.
Dioxinide furorate.
That's another one. No,
Humatin.
Humatin. Forget Humatin. Doesn't usually work very well.
It says Humatin is the luminal drug of choice since furamide is not available in the U.S. or Canada.
Is this a recent posting?
Only available from CDC. Well, it's Wikipedia. You never know.
No, Wikipedia. I wouldn't go to that one. I'd go to the PDR.
Yeah.
And in the PDR, you can get Iotaquinol. Okay, Iotaquinol is available. That's a luminal amoebicide and metronidazole at low doses. But now if you've got the abscess, you have to give very high doses of metronidazole. gives you a metallic taste in your mouth. There's some antabuse-like effects, too. So if you're a heavy drinker, or even a light drinker, you shouldn't drink at all when you're taking this drug. Otherwise, you'll get an antabuse-like effect.
Antabuse?
Antabuse.
What's that?
It's a drug that they give to alcoholics to keep them from drinking.
I see. Makes you throw up.
It makes you feel awful.
Sort of like Clockwork Orange, huh?
Right. Exactly.
I don't know who will get that.
A subset of our listeners will obviously get that one. So Entamoeba histolytica turns out to be a really bad infection once you've acquired it. And
there's no resistance to metronidazole.
Not that we know of.
Interesting. Now listen to your textbook here. So first it says,
now what do I say?
No, you said everything right. I'm just embellishing.
Okay.
So the brain abscess, nobody survives that.
Not really.
Because by the time you diagnose it, it's too late.
That's correct.
In the case of infection of the pleural cavity, that would be your
lungs,
where it goes to the diaphragm from the liver.
Yeah.
Quick aspiration combined with aggressive therapy has saved the lives of most of those, but they say it's rare.
It is.
A liver abscess should be aspirated in the following circumstances. 1, if there is no clinical improvement within 48 to 72 hours despite appropriate medical therapy.
Right.
2, for abscesses greater than 10 centimeters in diameter.
Can you imagine how big that would be?
Big. 3, when there is marked elevation of the diaphragm.
Yeah.
4, for abscesses in the left lobe.
Mm-mm.
And 5, when there is negative serology, which might raise suspicions of pyogenic abscess, which would be
for bacteria. Yeah.
Why the left lobe? What's that about?
Well, it's in an unusual place. You should be in the right lobe.
Right lobe facing you, or which way?
I think from the back. This is the right lobe. That's right.
Right hand, right lobe.
Yeah, that's right.
Got it. All right. Now, how do we prevent these infections?
Well, it's very simple, Vince. Just simply avoid contact with feces.
Well, if I go to a restaurant and the guy is infected and he's got my salad,
Vince is
what
baiting,
do I do?
He's baiting me again because this is not an easy one to avoid because you're totally you're a victim of circumstance, basically.
So what you're saying is just don't worry about it.
If you have eaten out every night of your life, you've dodged a bullet if you've never caught this one. Because the fact is that this is not that uncommon as in terms of a carrier state.
So eat contaminated food. How many? A week later, you're gonna you get the bloody diarrhea.
Not right away, but eventually.
And how long will it last?
Well, I mean, I think if your immune system is going to knock this thing out, and it takes a long time for that, remember, because the contact with the cells, you've got yourself a 3 or 4 week period of illness. Nobody lasts that long without going to a doctor if they can. If you've got blood in your stool,
you go to the doctor right away because you think you have cancer.
That's usually right.
Yeah. And then people are relieved that they have Entamoeba histolytica.
That's right. There's a test for blood in your stool that they can perform even if you can't see it. It's called a Guayak Test.
Yeah.
A test for the presence of hemoglobin. So.
Yeah, little cards.
That's right.
Yeah, so they give that to you for occult blood, and it's supposed to be for cancer, but if you have Entamoeba, it's going to be bloody too.
This is all true.
You bring it to your lab, and they go, "Oh, you have blood in your stool."
Yeah, right.
It's Entamoeba.
Exactly.
It's Entamoeba, but it's not...
It's not Dispar.
I don't remember the name. I have to look it up.
That's right.
Vaccines may soon be available based on recent encouraging progress made in the laboratory. Vaccinate children in hyperendemic zones. What would be the hyperendemic zones?
Well, I think you'd have to look at where the most unsanitary conditions exist. And you're presented with a big problem here, Vince. You're an expert on polio. That's the same transmission cycle as Entamoeba histolytica. The vaccine for polio would be the same usefulness as it would be for this one. So
are you saying that India would be a main place? Especially Uttar Pradesh?
I would.
Which has the last bit of polio.
And also in sub-Saharan Africa.
Wow.
Yep.
And that's not the main issue there.
No.
Malaria is much more of an issue.
Or AIDS. That's right. We've got a bunch of other problems. The biggest problems are lack of governmental attention to basic sanitary conditions. That's the basic truth of the matter.
Now, here at Columbia, did you teach this to the students?
I'm still doing that if they're listening.
Yeah, but this was one of the...
I certainly did. I did the 3 diarrheal disease-causing protozoans.
Because they would see it here in New York.
Yeah, well, sure.
All right.
Absolutely.
Anyone listening from the medical students? class here at Columbia?
I bet you they might be.
I don't hear anyone.
No, we got a few emails from them.
Yeah, Heather last time. She
That's
sent
right.
us a picture of a doodle she did.
That's right.
And it's posted, by the way.
Great.
Let's do some email.
I'm all for that.
Is that a good idea?
Yeah, absolutely. Let's correct some of these misconceptions that I've perpetrated on the public.
We all make mistakes, Dickson.
We do.
Too.
You know what? I'm not at all embarrassed over that.
This is the beauty of having a public forum.
That's right.
That there are enough people listening that if you make a mistake, they catch it.
No, hopefully that
great.
there's enough people listening that they'll catch it,
If you never get corrected, then no one's listening.
That's what it means.
Because no one is always right?
That is exactly right. You can't remember everything. And since you're reading my book and I don't have another copy in front of me, I can obviously...
Well, I use it to ask questions. Don't you think it's a good...
that's a great idea. No, I listen,
nice
Vince. Thanks. Again.
And as you know, I don't know any of this, so I can ask questions.
Right.
The first letter is from Bjorn.
Yeah.
Who writes, Hi, Vincent and Dickson. I want to correct the statement you made in the trypanosome episode. Apolipoprotein L1 in human blood kills only the subspecies Trypanosoma brucei, whereas the East African subspecies Trypanosoma brucei and the West African subspecies Trypanosoma brucei gambiense have evolved mechanisms to escape the lytic apolipoprotein. This means human infections with these two forms, if untreated, are always lethal. The good news is that through efforts spearheaded by the WHO, the number of estimated infected persons has been reduced to about 60,000 per year. WHO data from 2006.
Right.
Thanks for your edutainment podcasts. Bjorn, who is from Germany.
Hey, Bjorn, thank you for correcting something that I've misled the audience on. I think you might want to revise the number of people suffering from sleeping sickness in Africa. I think the number is higher than that now because of the displaced people that are moving around in places where they ordinarily wouldn't go due to civil unrest and war. So the last figure I read was that there's something like 400,000 people that are currently infected with sleeping sickness. So that's a lot of people. And, these are the poorest people. These are not the people that are in a position to get treated. So they're going to die. It isn't 100% fatal, but it's certainly very high. Okay, it's like rabies, by 80%, maybe even higher than that. There are very few things that are 100% fatal. Except perhaps life itself.
Life itself is 100% fatal. Everybody dies, Dickson.
I'm afraid that's true.
The next one is from Thomas.
Okay.
Who says, Greetings, professors.
That's a good start.
I originally got hooked on your show after discovering the treasures that awaited me in iTunes podcasts, specifically Quackcast, Dr. Chrislip, whose droll sense of humor is quite like mine. Eventually, I discovered TWiP, then as an added prize, TWiV. Since I am 71, I have seen the whole practice and concept of medicine change radically, as has almost everything else in my lifetime.
Right.
I have always had an interest in medicine, and back in the 70s even did medical histories for a free clinic run by volunteers, varying in education from MDs to interested amateurs like myself. Isn't it interesting, the gamut of people who write to us, Dickson?
It is.
We are really privileged to be able to get these kinds of letters.
I totally agree.
I've been watching the TV show that Professor Despommier works for as a consultant, Monsters Inside Me.
Yeah.
Unfortunately, the show reminds me all too often how so many of the people featured could have saved themselves a lot of time and discomfort if the physicians they dealt with had taken the time to take a medical history at the outset.
Yeah, that's true.
In fact, I think the first question any doctor should ask is, have you traveled to a foreign country, and if yes, when?
That's funny you should mention this, Vince, because Dr. Harold Brown, my mentor, quoted Shakespeare for this one, and he posed this to the medical students every year, and no one got it, except a couple of students did actually get it, and it's called Unde Venice. Unde Venice. And it's a quote from Julius Caesar.
How do you spell it? Unde?
UNDE. VENIS. Unde Venice. And he would say, does anybody know what the phrase Unde Venice means? And it's from Julius Caesar. Shakespeare, of course, was a Latin scholar, and it means, where have you been? So, to know about parasitic infections, where have you been?
Well, Harold knew.
Yep.
Who was the director of infectious diseases here for many years. He was famous for asking people, where have you been? And he could figure out almost
Harold knew was
perfectly.
a genius. He was a genius. And he and Harold Brown together was the institutional memory for infectious diseases for
Correct.
this place. And Glenda Garvey, we shouldn't leave her out of it.
And they're all three of them are gone.
They're gone.
And the youngsters are taking over.
Well, it's too bad that there wasn't podcasts in those days that we could have recorded them.
Can you imagine the three of them?
It would have been unbelievable.
Harold, I didn't meet Dr. Brown. I just had him for a lecture. And
I paled by
Glenda
comparison.
Garvey,
I must tell you, I paled by comparison to those people because they were not just hyper entertaining; they were hyper informative.
You pale, I evaporate.
Stop.
In fact. Yes, okay. Also, while it may seem wise when you hear hoofbeats to think horses, not zebras, as someone who has visited Tanzania, I always consider zebras as well as horses. That's good. Anyway, your show has allowed me, through listening to the same shows many times, to absorb some of the new concepts current in medicine today. Even though my mind is a poor shadow of what it was in my youth, I am still able to learn, just at a slower pace. I recently considered purchasing Mandel's 2-volume tome on infectious diseases. Oof. I put it in my Amazon shopping cart where it sat while I gave some serious thought to whether I wanted to spend almost $350
that's right.
to purchase it. I finally decided not to, but found to my ultimate pleasure that an Amazon shopping cart that has something in it and isn't acted upon in a reasonable time activates itself. I am glad it did, as I am really enjoying reading it online and in the printed version. Online access and search is an added feature if you purchase the book.
Huh.
I think back to when I was a youth, and there was a book Dickson might recall, as he is advanced enough in years, titled Not as a Stranger, an interesting novel that fascinated me as it was centered on the education that led to acquiring an MD degree.
Uh-huh.
How things have changed. Smiley. I had an older copy of is it Mandel's Sick book on infectious disease dating back to the early 1980s, and it amazes me how so completely a different approach there is to infectious diseases due to our current knowledge and emphasis on both evolution and genetics.
Boy, also.
Anyway, continue with the great shows and realize your audience may extend beyond the boundaries of listeners you might have thought interested in it. I'm always surprised by who
Great
listens.
Email.
By the way, Thomas,
Yes.
if you ever want a medical book, why don't you let us know, because we do have a bookstore here, and it has a lot of used books that the students sell at the end of the year. And
that's right.
We'd be happy to pick one up.
Yep.
I would be.
Yep.
But that's great. What was I going to add? Well, Dickson, he's 71. He's slightly ahead of you, but not much.
No, but not by much. I thought he was going to mention another book, which I will use as my pick of the week. A book that's currently out of date in press, I'm sure of it, and it's called With Gun and Camera Through the Alimentary Canal.
With gun and camera?
Through the Alimentary Canal.
Alimentary?
Alimentary.
Okay. Thomas sent us a link to his Flickr page. You know what a Flickr page is?
I have no idea whatsoever.
Flickr is a website where you put your pictures. You upload pictures.
Oh, okay.
It's full of thousands of photographers.
Okay.
So this is Thomas's photo stream.
Oh, wow. He's a gifted photographer.
Some birds here and a matador.
Nice.
Some soldiers here, 1960.
Thomas.
Oh, look, this Godzilla movie.
Did you make a living as a photographer?
It looks like he did, because these are quite nice.
Excellent.
I don't know if we should post this. Do you want us to post it, Thomas? I'll send you an email and ask you. But these are some gorgeous pictures. A tiger, look.
Wow.
Cape Buffalo. And let's see, he's got 2 pages. So
Goodness gracious. Mount Kilimanjaro with snow on it.
Mount Shasta.
Oh, sorry.
Near Weed, California. I was just there. I saw Shasta and Weed. We went through Weed, and my son had a reel. And here is Tom himself.
Here he is.
He is a wise old man. Not old. He's a wise man. Green jays. Look at this. Bugs.
Nice. Very nice.
I wonder if he fishes.
He looks like he spends a lot of time in nature.
I think we could put a link to this because he'd probably love to have a lot of traffic.
Sure.
Thank you, Thomas. Dan writes, Dear Vincent and Dick, first, let me say that your podcast is fantastic, very informative, and enjoyable to listen in the mornings during my commute. The reason I am contacting you both is actually less about the podcast itself and more in regards to the subject matter or how one gets into it. I recently finished my undergraduate degree and am interested in going off to public health school in a few years. I've been interested in the infectious disease aspect of community/international health for a while now, and most of my undergraduate degree focused on medical anthropology and infectious diseases, basic biostatistics, ecology courses, a survey of infectious disease biology, and a class on disease ecology.
Right.
I've been thinking about the graduate school path, and I know that the spectrum of eukaryotic parasites, particularly the protozoan-caused diseases, are what I would like to focus on.
Mm-hmm.
However, as most master's programs don't seem to have a particular parasitology concentration, with the exception of Tulane's MSPH, I was curious what advice you two may have for someone interested in entering the field. Go into epidemiology, environmental health, population biology. Any advice will be much appreciated. Thanks for your time.
Well, I think Johns Hopkins has a good international program also.
What about here?
Harvard. The School of Public Health at Columbia doesn't recognize this aspect at all.
Okay.
We're not involved in infectious diseases to any great degree, unfortunately. But Harvard, Johns Hopkins, UCLA, Tulane. I'm trying to think of anybody else.
No, he said Tulane is the one he knew about.
Yeah.
Parasitology component.
That's right.
So those that you mentioned. What about is there any? Are there any in California?
Yeah, it's UCLA.
UCLA.
Yep.
Berkeley doesn't have one.
Berkeley has an international health program that does include infectious diseases. I wouldn't count them out. I would investigate that.
Where could he go for more information?
That's very good. It's two sources. The American Society for Tropical Medicine and Hygiene would be a good resource.
Excellent.
And the American Society for Parasitologists.
All right. And the don't forget the American Society for Dicson Despommier. That's medicalecology.org.
Right.
Last email is from Scotty, who wrote, thought you guys might get a kick out of this. Why Captain Higgins is my favorite parasitic flatworm.
Uh-oh.
And he has a link, and he says, Dicrocelium Dendriticum, I salute you. What is Dicrocelium Dendriticum? Do you know?
I do know that, as a matter of fact. It's a very unusual fluke infection transmitted partially with the help of ants. And it's got some bizarre features to it. And it's a cattle disease, primarily.
So this is the oatmeal. This is a site which I just heard about.
Yes.
On a podcast. If you search oatmeal, you get this fellow's blog, basically. Not all the makers of oatmeal. And he often goes to conferences to talk about how he did this. But anyway, it's a comedy site, I suppose. Why Captain Higgins is my favorite parasitic flatworm. Captain Higgins is a liver fluke whose real name is Dicrocelium Dendriticum.
Yep.
And it's a little bit funny business going through all this life cycle and stuff. And it's kind of cute.
Well, it gets you interested. See, there's the ant.
The ant and how it's transmitted, the hosts.
Yeah.
And the life cycle, but all in funny business. So we will post a short.
Yeah.
We will post a link to that.
That would be good.
What do you think of that, Dickson?
I think that's great.
I will make a note here that was the last one we did.
As long as the science is accurate in the humor, it's a great way of delivering information to young people.
Was it accurate?
I haven't read it yet, so I can't honestly tell you, Vince. But what I did once for a summary of the research that we were doing with Racaniello was I wrote a play. And the play starred Trick E. Nella and and the host, Sal, which was called Beef Stroganoff. And I used the New York City playbill format and wrote the dialogue for it, and it's posted on the trichinella.org site. It was fun to write, and it included all of the aspects of the life cycle of the parasite as well. So
would that be edutainment?
It would be edutainment.
Excellent. Well, you can find TWiP at iTunes, of course, and you can find us at microbeworld.org/TWiP. Microbe World also has an app for your smartphone, for the iPhone, I believe only. You can find us there as well. If you're new to TWiP, please go over to iTunes and leave a short review, just a few sentences. Helps keep us on the front page of the medicine podcast section, and that's where we want to be, so more people find us and more people learn about eukaryotic parasites.
This is true. In fact, we promise to do more of these in the future than we have in the past.
We will. As always, we love to get your questions and comments. You can submit them on microbeworld.org/TWiP or twip@twiv.tv. Thank you, Dickson.
Welcome, Vince.
Dickson is at Despommier.com. No, not really. You never bought that domain name.
No, I never did.
Should have.
Never did.
Probably somebody owns it. Let's see, actually. Despommier.com. Coming soon. Someone owns it. They're parking on it. They want to sell it to you for thousands of dollars.
Well, I don't have that money. Sorry, Vince. I already own my name.
Dickson is at Racaniello.org, medicalecology.com, and verticalfarm.com. Media
Actually,
Empire.
it's medicalecology.org, too.
But I thought you got the com. You did get both, didn't you?
Yeah, probably.
Verticalfarm.com. Let's see what we got. It's the Vertical Farm Project. You got both, Dickson.
Okay. There you go.
The Vertical Farm, The World Grows Up, will be coming out soon.
Yep. Just got a copy of the book, actually.
It's a beautiful book.
Thanks, Vince.
Check it out. You have been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.